Good morning, children. Welcome back to our online classes. Let us continue with the chapter number 5, Principles of Inheritance and Variations. In our last video, we have studied about the deviation of Mendel's law. Today, we will start with rediscovery of Mendel's law, and then we will continue with chromosomal theory of inheritance. Though Mendel published his work on inheritance of characters in 1865, it remained unrecognized for several reasons till 1900. And some of the reasons are: communication was difficult, so his work could not be widely published. His concept of genes as stable unit that control the expression of traits and of the pair of alleles which did not blend was not accepted. His approach of using mathematics to explain biological phenomenon was new and unacceptable. He could not provide any physical proof for the existence of factors. In 1900, De Vries, Correns, and von Schermark rediscovered Mendel's result independently. Due to microscopy, they carefully observed cell division. This led to discovery of chromosomes. Now, let us study about chromosomal theory of inheritance. It was proposed independently by Walter Suter and Theodore Boveri. In 1902, they united the knowledge of chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. According to this theory, all hereditary characters must be with sperms and egg cells, as they provide bridge from one generation to another. The hereditary factors must be carried by the nuclear material. Chromosomes are also found in pairs like the Mendelian alleles. The two alleles of a gene pair are located on homologous sites on the homologous chromosomes. The sperms and eggs have haploid sets of chromosomes which fuse to re-establish the diploid state. The genes are carried onto the chromosomes. Homologous chromosomes synapse during meiosis and get separated to pass into different cells. This is the basis for segregation and independent assortment. Now, experimental verification of the chromosomal theory of inheritance. It was done by Thomas Hunt Morgan and his colleagues. He observed that the two genes under consideration in his experiments did not segregate independently as in the case of characters studied by Mendel. It led to discovering the basis for the variation that are produced during sexual reproduction. Morgan carried out various dihybrid crosses in Drosophila with the genes that were sex-linked, that is, the genes present on the X chromosome. Morgan selected fruit fly Drosophila melanogaster for his experiments because they could be grown on simple artificial medium in the laboratory. Their life cycle is only about 2 weeks. A single mating could produce a large number of flies. It has four pairs of chromosomes which differ in size. There was a clear differentiation of the sexes, that is, male were smaller and females were bigger. It has many types of hereditary variation which can be easily seen through low power microscope. That is all for today. In our next class, we will be studying about linkage and recombination. I hope you have understood the topic. Thank you.